Hey everybody, in our last video we discussed, or I discussed the structure of DNA, but I didn't talk much about hydrogen bonding between the nucleotides. So let's take a look at hydrogen bonding and what a hydrogen bond is. First, it may help to distinguish a covalent bond from a hydrogen bond. Now, a covalent bond, so that's a chemical bond that involves the sharing of electrons between atoms. For example, so let's say this is a nitrogen atom, and let's say this is a hydrogen. And if these are covalently bonded, they're sharing electrons. And I'm not going to diagram the electrons. I don't know how that sharing process works. I don't know if anyone really knows for sure how that process works, but they're sharing electrons. And when they share their electrons, they're covalently bonded. And I put this thick bond or stick between them to indicate that they are tightly bonded. Now, a hydrogen bond, so we can define that as a weak bond between two molecules resulting from an electrostatic attraction between a positively charged atom and a negatively charged atom. So electrostatic attraction, what does that mean? So it just essentially means that one atom is positively charged and the other one is negatively charged. So in this case, the hydrogen here, well, let's say it has a positive charge, a slight positive charge, and let's say there's an oxygen over here, and let's say it's covalently bonded to another atom, and it doesn't matter what it is. So this oxygen is negatively charged, and so what can happen is, these can be attracted to one another, and this is a hydrogen bond. Now these bonds are relatively weak compared to covalent bonds. And in these bonds, both types of bonds are important for the structure of a nucleotide. Well, the covalent bonds are important for the structure of the nucleotide. The hydrogen bonds are important for the process that keeps the two strands of a double-stranded DNA molecule together. So let me see, I have a diagram here because I don't think, because I'm posting these videos to YouTube, I don't think I can really take images from a textbook that's copyrighted and use them. So use some diagrams of nucleotides from a textbook. If those diagrams are copyrighted, I probably can't show them in this video here because I load these onto YouTube instead of uploading them to ReggieNet because I think YouTube is really convenient. So what I did was I just made my own diagrams here. And again, we don't need to memorize the structures. What we need to memorize, remember, is just the general structure of a purine with this bicyclic ring here, or these two rings here. So the purines have two rings, pyrimidines have one ring, and what distinguishes one purine from another would be these sort of side groups here. So adenine has this nitrogen here, for example, and guanine has an oxygen right here. Now, what I wanted to point out was, with this diagram, was that adenine pairs with thymine in DNA, and we can see the two hydrogen bonds here. So in each case, we have a hydrogen covalently bonded to a nitrogen. So there's one here, and there's one here. And those hydrogens are hydrogen bonded to an atom in the other molecule. So this one right here is hydrogen bonded with this oxygen, and this one right here is hydrogen bonded with this nitrogen here. So we have two hydrogen bonds formed between adenine and thymine. And if this was an RNA molecule, sometimes you can form a molecule between a double-stranded molecule that includes a single strand of RNA and a single strand of DNA. And in that case, we would just switch uracil for thymine, and the hydrogen bonding works similarly between uracil and adenine, because the uracil and thymine structures are very similar. So let's look down here at guanine and cytosine. So guanine and cytosine, now when they're paired, we can see 3 hydrogen bonds forming between them. Similar to the adenine and thymine situation, we have hydrogen covalently bonded to a nitrogen here, a nitrogen here, and a nitrogen here. And we can see hydrogen bonded to oxygen here, nitrogen here, and oxygen here. So we have 3 hydrogen bonds forming between guanine and cytosine. And in this diagram, you can see, okay, this is that number 9 nitrogen in the structure that is bound to the 1' carbon of the 5 carbon sugar. This one down here, the 1' carbon of the 5 carbon sugar. And this is the number 1 nitrogen in the pyrimidine structure. And we see a similar thing down here. The number 9 nitrogen bound to the 1' carbon of the 5 carbon sugar, and the number 1 nitrogen over here bound to the 5' carbon, bound to the 1' carbon of the 5 carbon sugar. Okay, so let's summarize that. So DNA is double-stranded. DS, double-stranded. In DNA, A forms 2 hydrogen bonds with T, and G forms 3 hydrogen bonds with C. Now, because DNA is double-stranded, we can see some, the double-stranded nature allows us to do some interesting things with DNA and heat. Okay, so here's my diagram of a standard DNA molecule. Now this is a tube right here, and let's say this DNA is in a tube of water. So we have a solution of DNA. Now, if that solution, let's say, is at 37 degrees Celsius, which is just a human body temperature, about the human body temperature. In that form, we can expect that the DNA will be double-stranded. And these right here represent the hydrogen bonds formed between the various nucleotides of the top strand and the bottom strand. So if we take that tube and we heat it to, let's say, 99 degrees. So 1 degree below boiling. Well, what happens is that the hydrogen bonds will be broken. And what we call this is when we break these bonds, we are denaturing the DNA. So we'll end up with 2 single-stranded DNA molecules. So here it was double-stranded, down here it is single-stranded. Now I'll specify that this is double-stranded DNA, and down here single-stranded DNA. And if we cool that, say we cool it down to 37 degrees, but we cool it slowly. So what can happen is the DNA strands can anneal. So the complementary bases down here can find the complementary bases up here because they're attracted to each other, right? And if we cool it slowly enough and give the bases time to find each other, the DNA molecules can re-anneal. And if we don't give it time, if we just cool it rapidly, then the DNA may fold in funny structures. But if we cool it slowly enough, I think most of the molecules can find the correct complementary match in this solution. So we'll call this double-stranded DNA. So we can denature DNA by heating the temperature to something like 99 degrees Celsius, and we can anneal it by we can anneal the two strands by slowly lowering the temperature to 37 degrees. Now, when we're denaturing DNA, what we're doing is we're breaking all those hydrogen bonds. Remember, we said the hydrogen bonds are weak bonds, and the temperature isn't high enough to easily break the covalent bonds between the nucleotides, but it is high enough to break those hydrogen bonds, and that's how we can make single-stranded DNA from a double-stranded DNA molecule. Now we can do something interesting with the DNA in UV light, and we can determine what is called the melting temperature, the temperature at which half the molecules in the solution are single-stranded and half are double-stranded. We can use UV light to determine the melting temperature of a DNA solution. And we can do that because DNA absorbs UV light. So for example, if we have a solution of DNA solution, and let's say we're able to shine some UV light on this and measure how much DNA it absorbs. And really how you could do that, you'd have some sort of cuvette like this, especially designed cuvette. And you'd add your solution in here. And these machines called spectrophotometers. You can shine UV light through, and there'll be a detector over here. So you can determine how much UV is being absorbed by the solution or the DNA in the solution with this machine. But we don't really need to know that. We just need to know that, okay, we have DNA in a solution and we are measuring the absorbance, and we can measure it, let's see, at different temperatures. So on the y-axis, I'll have UV absorbance right here, and down here we'll have temperature, and we'll say it's in Celsius. Now I'll start at 50 down here, 55, 60, and I'll go all the way up to 100. $95, 100. Okay, so let's say, well, let's go down to 45. Let's go a bit cooler. Now, we can start measuring the absorbance of the solution right here. And what we might see is, okay, an absorbance level of somewhere down here. Let's just say this is low. And let's say this is high. So the higher this little dot on the scale, the more UV that is being absorbed by the solution. Now, as we start increasing, say we're measuring UV at each temperature, what we'll see, we're increasing the temperature, what we'll see is that it starts absorbing more DNA as we get hotter. I mean, more UV light as we raise the temperature, until eventually it plateaus. So we may get something like this. Well, we will get something like this. So again, what we did was we took the solution, we measured the amount of UV light that was being absorbed by the DNA in the solution, and we saw that the amount that is being absorbed changed or increased as we increased the temperature of the solution. Now, so what's going on here? It turns out that double-stranded DNA absorbs less UV light than single-stranded DNA. So single-stranded DNA absorbs more UV light than double-stranded DNA. So what's happening is we're denaturing the DNA as we increase the temperature. So down here, say a certain percentage of the DNA in the DNA solution is denatured, but another percentage is still double-stranded. So we're absorbing this much UV due to the ratio of double-stranded to single-stranded DNA in the solution. As we increase the temperature, the amount of single-stranded DNA in the solution relative to double-stranded increases. So we're getting more UV light. So this inflection point, the point at which the rate of change in this diagram, it goes from increasing to decreasing. That's the melting temperature. The easiest thing to do here, we could also consider it the midway point for this little shift right here. So the point from where we have the lowest reading to the point where we have the highest reading, that's called the hyperchromic shift, or the hyperchromic effect. And what that describes is the phenomenon by which the amount of UV light absorbed by a solution increases as you increase the temperature. Now the melting temperature, the midway point between the hyperchromic shift, or this inflection point in this graph right here, what that is, that's the point at which 0.5% of the DNA is double-stranded and half of the DNA is single-stranded. So let me say this again. The TM stands for melting temperature. So DNA molecules, solutions of DNA have a melting temperature. And the word melting means the hydrogen bonds are coming apart. And the melting temperature of a solution is the temperature at which half the molecules are double-stranded and half the molecules are single-stranded. And TM is usually written like this, big T, little m. Now, okay, let's say the TM of this solution is 69 degrees here. 69 degrees Celsius. Now, what if I need a different color pen? Let me see. Or I need a pen. I think I have a red pen here. So let's say we have another DNA solution. Let's say this DNA solution was, say, DNA isolated from E. coli. And this DNA solution is DNA isolated from Streptococcus. Now, I don't know if this is true if we did this experiment with DNA from these different species of bacteria, but let's say we do this experiment and we see that the TM for Streptococcus here is 74 degrees. So what does that mean? How come, what do you think? Why would the TM of the DNA in this solution be higher than the TM of the DNA in this solution? So the reason is that this solution probably has a higher percentage, or the DNA in the solution has a higher percentage of GC base pairs. Now remember, GC base pairs have 3 hydrogen bonds, while AT base pairs only have 2 hydrogen bonds. So it takes more energy to break DNA that has a lot of GC, that has more GC base pairs. what's the right way to say this? Sorry, I'm butchering that. I'm butchering this delivery here. Okay, so it takes more heat to denature a DNA molecule that has more GC base pairs than it does a molecule that has less GC base pairs. Maybe I can diagram that for you. Let's take very simple DNA molecules with just 5. I made this too long here. Just 5 base pairs. And I'm going to make these 2 Gs. I hope you can see this in the video. I'm writing kind of small. So 2 Gs, a C, 2 Gs. And let's go 2 Cs. You know what? Let's make this an a T, and 2 Cs. And I'm going to put the hydrogen bonds here. 3 there, 3 there, 2 here, 3 here, and 3 here. Now let's have another molecule down here. And again, I hope you are not watching this on your phone, but watching it on a large computer monitor or a big screen TV. So here is our other molecule. And what we have here, I want to put 2 A's, let's say a C and 2 more A's. And down here, 2 T's, a G, and 2 T's. So I'm going to put 2 bonds, right? AT base pairs have 2 hydrogen bonds. And we can count up the number of hydrogen bonds between these 2 molecules here. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 H bonds. And here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 H bonds. So these molecules down here are being held together by only 11 H bonds. This one up here being held together by 14 H bonds, hydrogen bonds. So we can predict that we would need more heat to separate these single strands than we would these single strands because they have a higher number of hydrogen bonds holding them together. So, okay, so that's it for denaturing DNA, hydrogen bonds, how hydrogen bonds help hold the strands together, how the hyperchromic shift or what the hyperchromic shift is. DNA absorbs UV light. Double-stranded DNA absorbs less UV light than single-stranded DNA. We also learned about the melting temperature of a solution of DNA and what the melting temperature means. Essentially, half the DNA in the solution is double-stranded and half the DNA in the solution is single-stranded. And we learned why different DNA molecules will have or could have different melting temperatures. Okay, in the next video, I think we are going to start looking at replication, and we are going to start with Michelson and Stahl's experiments, which indicated that DNA is replicated with a semi-conservative mode, or through a semi-conservative mode of replication. Okay, see you in the next video.